Hi everybody, this is video 2 on cells. We're going to talk about membranes. So first thing we need to do is draw a phospholipid bilayer. So here is the polar head and there's those fatty acid tails. So we're going to talk about the outside of the cell, and then down here this will be inside of the cell. And so to make a bilayer, you're going to have to draw another. So here's another polar head and another 2 fatty acid tails. We'll make it a little bit larger, so keep drawing a few more. So we are creating that amphipathic barrier. This is only going to allow certain molecules to come in because there's a phosphate group on those polar heads, so that's very hydrophilic. And then the tails are just nonpolar hydrocarbon chains, so very hydrophobic. So the molecules that would be able to get through have to like that hydrophobic region in order to get in. So they've got to be small, they can't have charges, they're usually not polar, so we want non-polar small molecules. And if they are non-polar and small, they might be able to sneak their way right in or right out of a cell. That's called simple diffusion. It's going from an area of high concentration to low concentration, and it does not require any energy. All right, one more thing that you can find in that phospholipid bilayer is cholesterol. You remember cholesterol is a steroid, so it has 4 fused carbon rings, and it gets in between these tails right here. So you want to draw a couple of them like this in between the tails. And it makes the tails not so close to each other, and it makes it more fluid. And if you think about a cell, you want it to be fluid, you want it to be flexible so that it will be able to allow things in and out and so that it can move. All right, next thing I want you to add are membrane proteins. So again, we're going to draw our lipid bilayer, and we're going to add some proteins this time. So these are just phospholipids. A protein could be peripheral. That means it's only on one side. So draw a little blob like that. That's the peripheral protein. And you're going to have to make some more. I want them to be red. Here we go. All right, I'm going to have to draw some more phospholipids here surrounding it. All right, so you think about that peripheral protein and you think about what it might do. It's only on one side of the cell, so one of the jobs it might be is recognition, and your cells all recognize if they belong in your body or not. So you have like little markers, so it could be like, hey, this is a Nancy Mancuso cell, it's good, don't kill me. Hey, I'm another Nancy Mancuso cell, I'm good, don't kill me. And cells that don't have that special recognition protein, they're going to be noticed as non-self cells, and your immune system would attack them. All right, another kind of membrane protein is called integral. Integral means it goes the whole way through. So in both my peripheral and my integral, there's going to be regions that are hydrophobic and regions that are hydrophilic. So I'm going to take this purple here and I'm going to show, all right, any part of this protein in this area and any part of the protein in this area and this area, they must be hydrophilic. So think about those side chains. The side chains might have oxygen or nitrogen or sulfur, and they fold towards the outside because they're hydrophilic. There are other regions, I'll use this light blue color. This would have side chains that bend in because they're hydrophobic. So they probably have a lot of methyl groups. So they're going to go towards the middle where the fatty acids are in the lipid bilayer. So it's very important that you understand the structure of a protein to be able to understand how it would arrange itself as a peripheral or as an integral protein. All right, next thing we have to talk about are channels. Channels are proteins that are integral. They go the whole way through. So I'm going to draw a channel right here. It's got to be integral. And I'm going to make it look like it's a tube. And I'm going to make it have a gate on it. Many channels have gates. Now, these channels are great at moving molecules from areas of high concentration. So see how there's lots of them there? And just draw a couple over here. This is an area of low concentration. So if the gate is open, has to be open or they won't be able to move through, and usually your body has signals to open gates. If the gate is open, these molecules can move straight on through. We don't call it simple diffusion because they didn't get through the bilayer. They had to go through a membrane protein. So it's called facilitated diffusion because they're using the assistance of the membrane protein. It still does not require ATP. just like simple diffusion, because it's going from an area of high concentration to low concentration. Now, some examples. In your nerve cells, you have sodium-gated channels and you have potassium-gated channels. Those will be really important for moving ions. We know ions could not get through the bilayer, so this is how they get in and out of cells. Another example, in your kidneys. You want to get a lot of water out of your urine before it is eliminated from your body so that you don't dehydrate. So you have special membrane proteins called aquaporins, and they can move a lot of water at once. Water is one of those unique molecules that actually can make it through the lipid bilayer, but not a lot of them. So if you want to move a lot at a time, you need an aquaporin. All right, last thing here, pumps. So again, I'm going to have to draw a little bit more phospholipid bilayer because I have a new screen, but you might be able to just add it to your last diagram. So a pump takes on a unique shape. So I'm going to make mine like this, and that funky shape tells you it's a pump. And a pump is exactly as it implies. It's active transport. It has to actually use ATP to allow molecules to move. And the molecules exactly fit in these openings, this shape, and it moves things from areas of low concentration to areas of high concentration. So you might be thinking, if they already have a bunch of it, why do they need more? So think about a cell as like stockpiling. If it's calcium and it's like, but I want more calcium, or glucose and it wants more glucose. So if it's trying to move something against the gradient, it requires a pump. There's one that is very, very important that we will refer to multiple times this year. Those are proton pumps and sodium potassium pumps. A proton pump is going to move an H+, that's a proton. A sodium potassium pump is going to move sodium ions and potassium ions. All of those movements are against the gradient, therefore the name pump.